All right, we're going to take a look at the mechanism of how muscle contracts. On the molecular detail, we're going to go in and take a close look at actin and myosin and troponin and tropomyosin and how all of these play together. Before we go and look at the slides, I want to take a moment to draw this out and walk you through this process in painstaking detail, which should be your clue that this is an important process that you need to know step by step, every part of this process very, very well. So you'll want to make sure you study this hard so that you can know this material. We're going to start out with the role of calcium in muscle contraction. And for this, I've just drawn a diagram. Let me orient you to my drawing here. Each of these purple circles are my actin. So this is my G-actin in particular, and because it's bound to other G-actins, we have my actin filament. Okay. The molecules that are shown here in green, this is my troponin. And troponin is a complex of proteins. There's 1, 2, 3. This one over here, this green one, is called C-troponin. I don't really worry too much about those, so you don't really need to worry about that part of this detail. For now, just know that it's troponin, which is this complex of these proteins that work together. Now, troponin's job is it's a calcium sensing protein. That's what its purpose is. It senses calcium. It's a calcium sensing protein. And it involves these three molecules here. Now this orange fibrous protein shown right there, this is tropomyosin. And tropomyosin is, you can think of it as a competitive inhibitor. And what it's inhibiting is the binding of myosin to actin. So prevents binding of myosin to actin. Prevents. Okay, so it gets in the way. The actin binding site, if we were to kind of highlight this a little bit, that actin binding site would be found right here, down here at this bottom that I'm coloring in a little bit in yellow. That actually is the binding site where actin and myosin would bind together. So at rest, when our cells are at rest and we this would mean we're not generating tension. Okay, so the muscle fibers are not shortening, sarcomeres are not shortening, we're not generating any tension at rest. Okay. At rest, we would be in this arrangement with the tropomyosin blocking any type of myosin binding to that actin, troponin sitting on top there, and we have our actin filaments like so. When the skeletal muscle fiber experiences an action potential, and so recall that action potential, we get an action potential in the somatic motor neuron, acetylcholine released at the end, at the neuromuscular junction. We get a gradient potential generated by the activation of those nicotinic receptors. We get an action potential generated in the sarcolemma of the skeletal muscle, which changes the conformation of DHP, which activates the ryanodine receptor, which releases calcium into the cytoplasm, sarcoplasm. So when we have all of those things happening together, then calcium will bind. So calcium's going to swoop in here and bind to this binding site right here. This is my calcium binding site. And when that happens, we're going to get a chain reaction. Calcium binds to troponin, And you can see I drew this little red circle here. This is my calcium. I know it's hard to read that white writing, but this is my calcium. Okay. It binds to troponin, and it's going to change the physical conformation of troponin. Remember, conformation means shape. And so that protein or complex of proteins are going to change shape. And as is often the case, the change in shape of one protein will induce or cause a change in shape in the other protein. And in this case, that's true. The troponin shifts its conformation, and in doing so, it takes that tropomyosin. It's bound to the tropomyosin. It takes the tropomyosin and it pulls on that tropomyosin and drags it away from the binding site on actin. So now suddenly that myosin binding site is exposed, and we can now get the formation of these cross bridges that I discussed. And that essentially is the process by which calcium initiates muscle contractions. Really what it's doing is just exposing those binding sites so that myosin can get in there and do its job. Now let's look at how myosin works. And so I essentially just copied and pasted so we can get a fresh start. Here we have the conformation of the actin fibers when there is calcium present in the sarcolemma. Or sarcoplasm, sorry. So now we're going to take a look at myosin, and we're going to do some step-by-step processes. So first we're going to start with myosin as being in what we're going to call the starting position. At first, in the starting position, and let me draw it, I want to try to draw it the way that your figures show. So we're going to start here, and I'm going to have my myosin in this starting position, and it'll be shaped like so. Here's my myosin head, and I'm intentionally drawing it so that there is no contact between the myosin and the actin yet. Those binding sites have been exposed, and they're available. But at this point, what we have is this myosin fiber, or myosin head, and it is currently bound to a molecule called ADP and inorganic phosphate. So we're going to have this configuration in my starting position. Okay. Now, in order for this to bind, okay, to the myosin, all right, we're going to need my, actually, I apologize. Let me flip this around a little bit. We're going to put my starting position at more of a neutral angle, like so. Okay. So I'm going to throw my ADP and my inorganic phosphate in there. Okay. Now, in order for it to be ready to bind, my inorganic phosphate and my ADP are going to leave. And when that occurs, so we're going to kind of draw this arrow to suggest a change in time, what we're going to have happen is this myosin head is going to swivel into this next position here. Okay. And in this position, it will be loosely attached, perhaps, to that actin. And I'm going to call this the forward position. Any interaction with the actin at this point is fairly light. There's not a tight bond there, just kind of a loose attachment, and sometimes no attachment at all. Okay, but in this position, in this forward position, what we're doing is we're getting ready to generate some force. I'm going to quickly copy this so I can keep going with my diagram here, and we're going to move it down here. So we, again, representing just simply step by step these processes, okay? So moving that down here. The next part, or the next thing that's going to happen, is what we call activation of the myosin head. Okay, so we're going to put my myosin back here. Like so. And I want to draw it so it's not bound, so we're, oops, not what I wanted to do. We're just going to leave it there. Activation of the myosin head requires that ATP swoop in here and bind to myosin. So with ATP bound, this myosin head is now able to perform its catalytic function, which what it's going to do is catalyze ATP into ADP, an inorganic phosphate. And in the process of doing that, what is going to happen as we catalyze this ATP and inorganic phosphate is that we are going to once again move that myosin head. Let me go back up here and still this one. Coming back here. Okay. And I'm just going to add a few extra ones just so that I can have them when I need them. Okay. Alright, so once again, we're going to take this myosin head and we're going to move my myosin head. Come on. Try this again. Move this up so I have room. There we go. When ATP binds my myosin head, myosin is going to swivel into what we call the cocked position. So I'm going to very briefly in dotted lines show you where it was. Okay, and then the cocked position is actually back here. And in the cocked position, we get a tight bind to, or a bind, let me rephrase that. At first it's a loose attachment. We have ADP, because remember in this forward position we had ATP, but now ATP is hydrolyzed into ADP, an inorganic phosphate, and in the process of doing that, the energy that is released when we break those chemical bonds causes a change in that myosin head, in the position of that myosin head, and swivels it into my cocked position here. In the cocked position, it loosely attaches to the actin. The next process, or the next step, is for this inorganic phosphate to leave. And so if we take a look quickly at that process here, okay, when that inorganic phosphate leaves, the myosin head attaches much more strongly to the actin filament, okay, more strongly, so we get this very strong, robust attachment here, okay, nice strong binding here, and once the phosphate leaves, we still have my ADP left, but the phosphate has left, and that leaving of the inorganic phosphate triggers what we call the power stroke. And the power stroke causes, let me actually move that and write it over on the side here so we have more room. This is my power stroke. And that power stroke causes this to move back into this forward position. But the thing here is when it moves backwards, it actually is taking, if we can kind of label these as 1, 2, actually let's go back up here, label this as 1, 2, 3, and 4. When this moves into the cocked position, what it actually is going to do is shift everything forward. And so this whole actin filament is going to move so that 1 actually moves. I'm going to draw a dotted line to show you where it's going to move. So this is where 1's new position is, which would make this 2 and this 3 and this 4 and this is 5, which I haven't drawn up here, but it would be tugged in with it. And so I hope you're seeing how the whole thing would shift down. Now during the power stroke, at some point, and a lot of people will argue about when and where this is, at some point ADP is also lost. Now some textbooks will say that ADP is not, that ADP remains bound to the myosin until ATP takes its place. Some will say it's lost in the midst of that power stroke somewhere. I'm not super picky about that information because honestly it's still to be determined. It's hard to figure out exactly when that ADP leaves because we don't have the technical ability to zoom into the atomic level or the molecular level and witness this in action. The best we can do is essentially freeze these at any given point in time and using X-ray crystallography and so forth try to image and figure this out. So we know that ADP is at some point lost during that power stroke, either at the very end of the power stroke or in the middle of the power stroke somewhere, either before or after we're in the forward position. Now in this forward position, it's a little bit different. Let me kind of go back up here and point out that it's a little bit different. Okay, so here we're loosely bound. This was my starting point. Loose binding or perhaps no binding at all. If we go down here though, when calcium is present, when we move into that forward position and ADP is lost, okay, let me move this out of my way. Okay. We end up having what is known as rigor state. Okay. And in rigor state, that myosin remains very tightly bound to the actin. So this is rigor state. And we would still, of course, let me kind of pencil this in really quickly. Of course, 1 is going to be right here. So we're looking at 1, 2, and 3 having been dragged into place. Now, rigor state is a tight attachment, and it is associated with a term that you will recognize, rigor mortis. Now, just because your myosin is in rigor state doesn't mean you're in rigor mortis. Rigor mortis is a condition that occurs after death. And it is in fact when your skeletal muscle freezes into this rigor state. And the reason it happens is after you die, you're no longer breathing in oxygen. Without that oxygen, the mitochondria, it takes a little bit for your cells to catch up to this idea of being dead. The body is dead, but cells themselves can actually remain viable in some cases for some time later. This is actually why organ donation works, is because you have a certain window of time that you can harvest those organs and have them still be viable for transplant. But rigor mortis then would occur maybe 12 to 24 hours after death ensues. because you're no longer bringing in the oxygen, and because you don't have oxygen, your mitochondria can no longer make ATP. And in the absence of ATP, okay, your skeletal muscles get stuck in this rigor state. Let me tell you how you break rigor state. Rigor state is broken, so you break rigor state by having ATP bind to that myosin. Once that occurs, rigor state is broken. So let me jot that down. Rigor state broken when ATP binds myosin. Okay? And so you'll want to make sure you understand that and know that. And then of course, once rigor state is broken, we're actually right back where we need to be to go through another Crossbridge cycle. This, drawing it again, ATP binds to myosin. Myosin is going to hydrolyze that ATP, and when it does hydrolyze that ATP, it will move from this position here to, oops, yeah, we'll draw it there. There you go, the cocked position once again. And that occurs when the chemical reaction occurs that hydrolyzes ATP into ADP in inorganic phosphate. And at that point we're going to repeat. Phosphate is going to leave. Phosphate will bind to the myosin, myosin will bind tightly to the actin as phosphate leaves, and then it will execute the power stroke and ratchet everything forward one position. Now, it might not seem like, if you think about this, you're like, oh my gosh, it's going to take forever for these muscles to contract. But recall, if you will, that we have many different myosin heads attached to the filaments at the same time. So going back to the PowerPoint, we can actually watch this process or look through this process. And if we look at these figures, you can see, so if you study the figure on the left, what we're actually looking at in this case is the role of troponin in moving that tropomyosin. Let me kind of pick a different color to highlight it. So here's my troponin molecule, and they didn't draw the complex, they just drew a circle, which is totally fine. But as it binds to this calcium, you'll see that my tropomyosin is moved out of the way, and that exposes the actin binding sites so that myosin can bind. Once that myosin binds, you'll notice here that I have my inorganic phosphate leaving, and it's this leaving of this inorganic phosphate that allows for this tight attachment to the actin. When we, before that phosphate leaves, the best this can do is kind of a loose attachment. Okay? That's the best we can do. But once that phosphate leaves, we can get this strong attachment, and then that actually executes the power stroke, which would cause this to ratchet forward so that we move the actin, and in this case that actin, everything would shift in this direction, and so we would see it moving. If we could watch this, if we could actually see a video of this, and there's tons of animations available, but if we could watch it in real life, we would essentially see that actin moving with the myosin. And so that looks at how the calcium involved or is used to change troponin and to cause that tropomyosin to move out of the way. And then here, if we actually look at this Crossbridge cycle, we can actually see two components of it together. And let's zoom in on this a little bit so you can see here it's going to start in a little bit of a different spot, but it's a cycle. So cycles, it's fair game to start wherever you want in the cycle because everything is circular, right? So here we actually start with ATP right here bound to my myosin head. And notice that when ATP binds, look at that, the event The process of ATP binding changes the shape of myosin so that it can no longer bind actin. Okay, so it's not bound to actin. When ATP is hydrolyzed into ADP and inorganic phosphate, do you see how we moved into the cocked position right here? And that action of moving is caused by that chemical reaction. You know, if we were to write that chemical reaction, ATP to ADP and inorganic phosphate. That's the chemical reaction that occurs. It's this chemical reaction that moves us into the cocked position. Once we're in the cocked position, okay, this actually kind of flips things over a little bit. I wish it didn't, but unfortunately that's just how the figures are. So, but once we're in the cocked position, when phosphate leaves, so it exits, we go from our cocked position and we say that we generate a power stroke, and that power stroke moves us back into this starting position. Now in this particular figure, or forward position, I think I like forward position better, this forward position, and this particular figure shows ADP is still being bound. And that's okay, because in some like I said, in some textbooks it shows us it's still being bound at the end of the power stroke. In other textbooks it's going to show ADP leaving during the power stroke. What we do know for sure is that ADP is at least knocked out of position by ATP. Okay? Now, so in this particular position, if we go further down and we take a look at this, this is a figure from a different textbook, so we're flipped upside down. I apologize about the different orientations. Sometimes the best pictures are not, well, we just end up with different positions. Come on. Alright, it's being difficult not letting me zoom in. Let's try one more time. Nope. Okay, so you're just going to have to kind of do your best to take a look at this. So when my ATP, so in the forward position, pay attention closely to this binding. You see this binding right here? It's tight. Tight, tight, tight binding. And that's my rigor state right here. This is also rigor. This is rigor. This is my rigor state. And that myosin will remain bound to actin until ATP sweeps in and binds myosin. The moment that ATP sweeps in and binds myosin, you can see, if I kind of color code that in, that our binding has been lost. So the binding of ATP causes the myosin head to deattach. And so that's a necessary step for this process. Now once that myosin has deattached, now we're once again ready to hydrolyze that ATP, move back into the cocked position. Phosphate would then leave, causing us to execute the power stroke, and then ADP would be replaced by ATP, and so forth. And so this together is what we call our Crossbridge cycle, which this time it let me zoom in, which is awesome. And you can see this, and really we'll start at the resting fiber. What you're not seeing here, you're not seeing the tropomyosin blocking the way there, so I'll just go ahead and scribble that in and say, hey, you know, tropomyosin is in the way. Troponin is there too. It's not drawn in this particular figure, but it would be there, so we'll just throw in a few troponin molecules there. And so in my resting state, this is essentially what we see. But then when tropomyosin is moved out of the way, we're ready for us to begin by having myosin attached to actin. Okay, so myosin binds. My ADP, my inorganic phosphate leaves, causing it to execute the power stroke. And at some point, ADP leaves. This is showing it as being after. It could leave here too. We don't know for sure. And now we've got this vacant spot, and we've got ATP ready to sweep back in, bind that spot. As soon as ATP binds, so this is rigor state, rigor is broken. And then once rigor is broken, we're ready to hydrolyze that ADP into inorganic phosphate. And this time, as long as calcium is present in the cell, that myosin, tropomyosin, is not in the way anymore. We've got it instead, up here where it's out of the way. And so now we can continue the process. ATP is hydrolyzed into ADP and inorganic phosphate, right? And so that's my chemical reaction there. And we're right back into the same situation where we're now ready to form a cross. We go back into the the cocked position. And then we're going to have phosphate leave. We execute the power stroke. We've got rigor state again. We're going to break rigor state with another ATP. And this process will continue as long as calcium is present in the sarcolemma. This brings us to a vocabulary term or phrase that you'll want to know, and it's called excitation contraction coupling. And that essentially is a fancy phrase to describe exactly what we just walked you through. Excitation contraction coupling is the process that links the action potential in the skeletal muscle to the events that are involved in skeletal muscle contraction. So action potential releases the calcium, and calcium stimulates the contraction by making sure that those myosin binding sites are available on actin. And so calcium is the link to this entire process. Excitation contraction coupling would not occur without calcium. And so that's super important, super critical that you need to understand that. And so that's essentially the process of excitation contraction coupling, which involves calcium binding to troponin, moving tropomyosin away, activation of the myosin head by binding ATP, myosin head binding the actin, and so forth. So please, please spend some time really looking at this and understanding this, and know it well. These types of main concepts can sometimes show up as essay questions. Even if they don't show up as essay questions, they frequently end up as more detailed, thought required type of multiple choice questions. And so you'll need to know this really well. My advice would be to draw it out multiple times. Just draw, 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 draw. And each time you draw, to list step by step each step and what happens in each step and what each phase is called. This is the power stroke. This is the cocked position. And know what those are. This is rigor state, so that you can answer questions about any part of this on the exam. If I ask you what happens, what is rigor state? How is rigor state broken? Which of the following steps happens before we end up in rigor state? Those are the types of questions that I could ask in a multiple choice true-false question type phrase. For the essay questions, I could ask about drugs that bind up calcium, or maybe drugs that reduce ATP production and try to get you to think about how that would affect the myosin crossbridge process.